antimicrobial agent is defined as a natural or synthetic substance that kills or inhibits the growth of microorganisms such as bacteria, fungi, and algae. They are classified into several categories based on their mechanisms of action, such as disrupting the plasma membrane, inhibiting cell wall, protein, DNA, and RNA biosynthesis. Antimicrobial resistance occurs naturally via sporadic genetic mutations. This scenario exerts a positive effect in natural selection that those without the resistant genes will be killed, leaving those who possess them to thrive. Antimicrobial resistant genes could usually be found within the plasmid that can be transferred through conjugation, and sometimes within the chromosomal DNA, which can be transferred through transformation. In rare cases, resistant genes could also be inserted by a virus through transduction. Organisms with antimicrobial resistance could be found everywhere in the environment and can transmit the resistance from person to person or from animals to humans. Nevertheless, antimicrobial agents are widely used in agriculture to maintain animal health in green traffic. However, overuse of antimicrobial agents has speeded up the emergence of antimicrobial resistance microorganisms, and this has become a major concern due to the presence of multi-drug resistant microorganisms in every level of the food chain. As a consequence, antimicrobial resistance could be transmitted to the consumers at higher levels. Besides, overprescriptions of antibiotics over the counter has also caused the emergence of superbugs that are resistant to almost all antibiotics. In the United States, the epidemic of antimicrobial resistance is closely monitored by the National Antimicrobial Resistance Monitoring System, NARMS. It is a collaboration among Centers for Disease Control and Prevention, CDC, the U.S. Food and Drug Administration, FDA, the United States Department of Agriculture, USDA, as well as state and local health departments in all 50 states. It was established in 1996 after an expert panel convened by the FDA in 1994. The main objectives of NARMS are: monitor trends in antimicrobial resistance among enteric bacteria from humans, retail meats, and animals, disseminate timely information on antimicrobial resistance in pathogenic and common cell organisms to stakeholders in the United States and abroad to promote interventions that could reduce resistance among foodborne bacteria, conduct research to better understand the emergence, persistence, and spread of antimicrobial resistance, provide data that assist the FDA in making decisions related to the approval of safe and effective antimicrobial drugs for animals. NARMS advances food safety and public health in various ways. It provides data on the emergence of drug-resistant enterobacteria, movement of bacterial populations among humans, food animals, and other sources, genetic mechanisms underlying resistance, and risk factors for outcomes of resistant infections. These data are essential for ensuring that antimicrobial drugs approved for food animals are used in ways that are safe for human health and to limit the spread of resistance. Under the microbiological research conducted by NARMS, the DNA of the clinical isolates are sequenced through whole genome sequencing using Illumina MiSeq. It implements next-generation sequencing, also known as massively parallel sequencing and deep sequencing. The Illumina next-generation sequencing comprises 4 distinct steps: library preparation, cluster generation, sequencing, and alignment and data analysis. Library preparation. The sample DNA or cDNA is broken into smaller pieces through random fragmentation. Oligonucleotides known as adapters are then ligated at the 5' and 3' ends of each fragment. Each adapter has 3 distinct regions that serve different functions, such as sequencing binding site, indices, and a complementary sequence that can hybridize to the oligos on the flow cell. Otherwise, the library preparation process could be efficiently shortened by combining sample fragmentation and ligation under the same reaction known as tegmentation. Adapter ligated fragments are then separated into single strands by incubating them with sodium hydroxide. Cluster generation. A flow cell is made of a glass slide with multiple lanes. Each lane contains a lawn with surface-mount oligos that have 2 different sequences. Single-stranded fragments are then added into the flow cell, allowing the complementary adapters and oligos to bind together. Unattached fragments are then washed away. The attached fragments are amplified through a polymerase chain reaction to form the complementary strands. Then, the double-stranded fragments are once again denatured and the unattached original fragments are washed away. The free ends of the fragments build bridges by binding to another complementary oligo on the flow cell. Through bridge amplification, the single-stranded fragments are amplified to form double-stranded fragments. They are denatured to form two separate single-stranded tether onto the flow cell. The reverse fragments are cleaved and washed away. The reaction repeats several cycles to form clonoclusters with identical sequences. The forward fragments are ready for sequencing once the cluster generation is completed. Sequencing. Primers bind to the sequencing binding sites on adapters. One fluorescently labeled nucleotide is added at a time to the template strand by DNA polymerase based on the sequence of the template. The pairing of the fluorescently labeled nucleotides to the template strand is governed by the Watson and Crick base pairing of the DNA. Once the fluorescently labeled nucleotides are excited by a light source, they give off signals at different wavelengths. When a fluorescently labeled nucleotide is added to the template, elongation of the DNA is terminated until the blocking group is removed from the nucleotide. This is known as the reversible Terminator-based method by Illumina SBS technology. The wavelength of the signal tells the base of the nucleotide, while the intensity of the signal tells the number of bases added. This entire process of sequencing is known as sequencing by synthesis. Data analysis. The overlapping sequence of the fragments are arranged and processed to obtain the sequence of the whole genome. The sequence of the sample is then analyzed and aligned for comparison with a reference genome. Through the analysis and alignment, any alteration to the sample such as single nucleotide polymorphism, insertion and deletion could be detected precisely. Whole genome sequencing of the bacterial isolates allow detection of the genes conferring resistance to antimicrobial agents. The bacterial isolates then undergo antimicrobial susceptibility testing to determine their resistant phenotypes or resistant profiles. The genotypes of the bacterial isolates are then compared to their resistant phenotypes to confirm antimicrobial resistance. The genome sequences of new bacterial isolates are uploaded to CDC PulseLab or FDA Genome Tracker. NORMS laboratories and databases house specimens and data for more than 200,000 bacterial strains from humans, food animals, and retail meats. In the case of outbreaks, the bacterial strains are cultured for whole genome sequencing and compared to the sequences from the databases. If the sequences are matched, the origin and transmission of the outbreaks could be traced and appropriate measures could be taken immediately. Here comes the end of the animation. The theme of this animation is inspired by a YouTube channel called Kurzgesagt, and everything in this animation is hand-drawn by our members. Thanks for watching and I hope you liked it.